Okay, so this one, glucose to glucose 6-phosphate, that's glucokinase, hexokinase, that's irreversible. Pyruvate to PEP, that's pyruvate kinase, right there, is irreversible. And this one, fructose 1,6-bisphosphate, PFK1, is irreversible. So you have 1, 2, 3 irreversible steps. The rest are reversible. So you have to bypass these steps. Okay. All right, so let's start with pyruvate. Because this step is irreversible, you have to come up with something else. In fact, you're going to need 2 enzymes to convert pyruvate back to PEP. Okay? So this is going to require 2 enzymes, and we'll start here. So we're going to start with these 2 enzymes that are required to convert pyruvate back to PEP. So first of all, I want you to appreciate this important thing here. Pyruvate, if it's produced from glycolysis, let's say, where is it? In the cytoplasm. It can actually be produced, I want to point out to you, from other sources, like alanine, the amino acid, and give you pyruvate. Let's say you had some pyruvate in the cytoplasm, or lactate that went to pyruvate in the cytoplasm. Before pyruvate can undergo gluconeogenesis, and note, this is the direction of gluconeogenesis, it's the reverse. You have to bring in pyruvate into the mitochondria, and there's a specific transporter there that brings it in. So let's say you bring in pyruvate into the mitochondria. That's where gluconeogenesis begins. In the mitochondria, you're going to convert pyruvate to a molecule called oxaloacetate by adding a CO2 to it, a carboxylic group, from 3 carbons to 4 carbons. This is the first enzyme here in the mitochondria called pyruvate carboxylase. Converting pyruvate to oxaloacetate. The rest of gluconeogenesis is going to be in the cytoplasm. Okay? So you have to bring that oxaloacetate out. But the mitochondria have this impermeable membrane. So before you can bring that oxaloacetate out, you have to convert it to something else. And you convert it to malate. You reduce oxaloacetate to malate. Malate can be transported. There's a specific transporter for malate that will take it out to the cytoplasm. But let's focus first on this enzyme here, the first one, pyruvate carboxylase. We've already covered that. It's the first bypass to bypass the pyruvate kinase step, okay, in glycolysis. This enzyme is present only in liver and kidney and not in muscle. That's why muscle will not have gluconeogenesis. 2 important things about this enzyme. You're going to need ATP, CO2 as for the reaction, and the coenzyme vitamin biotin. Okay? So here's CO2, here's ATP, and this is the enzyme, coenzyme biotin needed for pyruvate carboxylase. And that, again, will occur in the mitochondrial matrix, okay, to form oxaloacetate. This is a little bit too much detail, but it's just to show you, really, you don't need to know all the details, except here's biotin attached to the enzyme. And it is the carrier of that CO2 that eventually gets incorporated into the oxaloacetate. So that's how some of these coenzymes function. They play a key role in the transfer of what's being added here. In this case, it's a CO2. So all you need to remember is you need biotin for pyruvate carboxylase, you need CO2 and ATP. So you form oxaloacetate in the mitochondria. Like I said, the rest of gluconeogenesis, going up back to glucose, is going to occur in the cytoplasm. So you have to transport oxaloacetate. This is the mitochondria, if you're not sure. So you have to transport that out. You have to convert it first to malate before it can be transported. One of the most important allosteric regulators of pyruvate carboxylase, that enzyme we just covered, is acetyl-CoA. Small molecule, typically produced from beta oxidation of fatty acids in the fasting state. So think about it. This high amount of this molecule will activate pyruvate carboxylase allosterically. You should know what allosteric means. It binds to the enzyme at a separate site and activate the enzyme from the catalytic site and activate the enzyme. Okay? So once you produce oxaloacetate, we convert it to malate by malate dehydrogenase. Malate is then transported out to the cytosol, where the rest of the enzymes required for gluconeogenesis will be. So the rest of the way, it's all going to occur in the cytoplasm. So oxaloacetate, malate, once malate is in the cytoplasm, it is going to be reoxidized to oxaloacetate. Okay? In the cytoplasm. The malate conversion to be able to transport it out of the mitochondria. Once in the cytoplasm, it's reoxidized by the same enzyme, cytosolic malate dehydrogenase, gives you oxaloacetate. Oxaloacetate now is going to be converted to PEP. So this is the second part of that bypass. Really, we're trying to make pyruvate, PEP from pyruvate, so we need two enzymes in succession. Pyruvate carboxylase was the first one to produce oxaloacetate. The second one is called PEP carboxykinase. And that converts oxaloacetate to PEP in the cytoplasm. So that's oxaloacetate. PEP carboxykinase uses energy from GTP to produce phosphoenol pyruvate. Okay, so this is kind of an energy utilizing step, because this is a high energy molecule we are producing. So in order to form PEP from pyruvate, you need in succession pyruvate carboxylase, then which is in the mitochondria, then PEP carboxykinase, which is in the cytosol. After you transport malate out of the mitochondria and then reoxidize malate to oxaloacetate. Okay, so this is again the big picture. This is the first enzyme. Here's the second enzyme. Okay, this is PEP carboxykinase, and this is pyruvate carboxylase. This one is allosterically regulated, activated by acetyl-CoA. So know the location, know what they're producing. Okay? So if you produce PEP here from PEP carboxykinase, now you can go backwards in gluconeogenesis in glycolysis because all of these are reversible.